Hello everyone, I am Nelly B.S. Lloren and I will be presenting the DNA replication. Here is the outline for our topic for today. We have importance of DNA replication, site of DNA replication, proposed models of DNA replication, steps of DNA replication, and compare the process of prokaryote and eukaryote DNA replication. Every cell in an organism has a particular lifespan. They don't last forever. Cells are born, grow and do their jobs, divide, and finally create two new daughter cells. The lifespan is cyclical, and with every new generation, the cells that are formed carry out the functions of the parent cell because they are given each an exact copy of the parent cell's DNA. The cyclical lifespan of a cell is called the cell cycle. This figure shows the cell cycle, and it consists of 5 phases. In 3 of the phases, shown in blue, the cell grows and prepares for division. Cell division, shown in green, occurs in 2 phases. During which phases do cells have duplicated chromosomes? During the synthesis phase, the DNA in each chromosome is copied. And during mitosis, the chromosomes are packaged in their most highly condensed form. Every time a cell goes through a cycle, it must duplicate its DNA so that when it makes new baby cells, they both have the same exact DNA. It happens in the S phase. In this section, we are going to explore the process of DNA synthesis, or also called DNA replication. This process is extremely precise and incredibly important. So what's the different about these cells? So as you can see, the blue chromosomes that are directed by the arrow are identical strands of DNA. There are 2 copies formed in replication because each are destined for the 2 identical new cells that are formed, called daughter cells after the cell divides. Identical DNA must be synthesized in order to achieve this. And this process of DNA synthesis is called replication. The elucidation of the structure of the double helix by James Watson and Francis Crick in 1953 provided a hint as to how DNA is copied during the process of DNA replication. Separating the strands of the double helix would provide 2 templates for the synthesis for new complementary strands. But exactly how do new DNA molecules were constructed was still unclear. Here are the 3 proposed models of DNA replication. We have the conservative model, semi-conservative model, and the dispersive model. In the conservative model, the parental DNA strands remained associated in one DNA molecule, while the new daughter strands remained associated in the newly formed DNA molecules. In the semi-conservative model, parental strands separated and directed the synthesis of a daughter strand, with each resulting DNA molecule being a hybrid of a parental strand and a daughter strand. In the dispersive model, all resulting DNA strands have regions of double-stranded parental DNA and regions of double-stranded daughter DNA. Matthew Meselson and Franklin Stahl devised an experiment in 1958 to test which of these models correctly represents DNA replication. The Meselson and Stahl experiment. They grew the bacterium E. coli for several generations in a medium containing a heavy isotope of nitrogen, 15 that was incorporated into nitrogenous bases and eventually into the DNA. This is labeled the parental DNA. The E. coli culture was then shifted into a medium containing nitrogen-14 and allowed to grow for one generation. The cells were harvested and the DNA was isolated. The DNA was separated by ultracentrifugation, during which the DNA formed bands according to its density. In the figure here, Meselson and Stahl experimented with the E. coli, grown first in heavy nitrogen-15, then in nitrogen-14. DNA grown in nitrogen-15, the blue band, was heavier than DNA grown in nitrogen-14, the red band, and sedimented to a lower level on ultracentrifugation. After one round of replication, the DNA sedimented halfway between the nitrogen-15 and nitrogen-14 levels, ruling out the conservative model of replication. After a second round of replication, the dispersive model of replication was ruled out. These data supported the semi-conservative replication model. As a result of these experiments, we now know that during DNA replication, each of the two strands that make up the double helix serves as a template from which new strands are copied. The new strand will be complementary to the parental or old strand, and the resulting DNA molecules have the same sequence and are divided equally into the two daughter cells. The Meselson-Stahl experiment is also known as the most beautiful experiment in biology, since it enabled researchers to explain how DNA replicates, thereby providing a physical basis for the genetic phenomena of heredity and diseases. So the replication of DNA follows the semi-conservative model, where each strand in the DNA double helix acts as a template for the synthesis of a new complementary strand. Here are the steps of DNA replication. The location at which a DNA strand begins to unwind into two separate single strands is known as the origin of replication. And why is this the particular origin? Because there are particular nucleotides in this region which are highly concentrated, and it is adenine and thymine. It's adenine and thymine rich area. Why would I pick adenine and thymine as the area I really want to target as compared to guanine and cytosine? Yes, because adenine and thymine has 2 hydrogen bonds, while guanine and cytosine has 3 hydrogen bonds. And it's gonna be easier to break 2 hydrogen bonds than to break 3 hydrogen bonds. And this area is better suited to be separated because there are only 2 hydrogen bonds, and it's gonna require less energy to break those hydrogen bonds compared to guanine and cytosine. So always remember that in eukaryotic cells, there are multiple origins of replication. So we have another origin of replication here and another origin of replication here. Question: what type of structure is going to bind onto these areas and help to break the bonds between adenine and thymine? Okay, there's a really interesting protein called pre-replication protein complex. This pre-replication protein complex, it binds to the origin of replication and separates the adenine and thymine nitrogenous bases. Due to the previous process, we can now form a replication bubble. These are the nitrogenous bases, and we're gonna expose it now. These were once connected nicely together, and they're kind of vulnerable. When you separate them, they want to reanneal with one another so badly. And what is this protein that really helps to protect these vulnerable separated parental DNA strands? This protein is called the single-stranded binding protein. What is the purpose of this single-stranded binding protein? One of the functions is that it prevents the parental strands from reannealing or reconnecting from one another, and they act as a barrier and protect the parental strands from enzymes that want to break the phosphodiester bonds called nucleases. Once you form the replication bubble, you form the two end here. And these two ends that looks like a Y is the replication fork. The DNA helix unwinds like a zipper as the bonds between the base pairs are broken. The enzyme DNA helicase is involved in breaking these bonds. The DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands to make a new DNA. There's a problem that happens as DNA helicase is going through and unwinding the DNA constantly in front of it. It bunches up the DNA distal to it from that replication fork, and it creates these things called supercoils. Why are supercoils bad? If you bunch up the DNA in front of it, it's going to impede the helicase from continuing to unwind the DNA. It's going to face a lot of restriction, so it will just keep getting bunched up until you relieve those supercoils. So we need enzymes that can come in there and fix these supercoils where the DNA is tightly wound. And these enzymes are called topoisomerases. Topoisomerases have three types: type 1, 2, and 4. The type 1 and type 2 is present in eukaryotic cells, while type 2 and type 4 is present in prokaryotic cells. Type 1 topoisomerases does not require ATP, while type 2 and type 4 topoisomerases requires a lot of ATP. The major function of the topoisomerase 1 is to relax supercoiled DNA and alleviate the DNA helical constraints. While topoisomerase 2, these are topoisomerases that cut both strands of DNA helix simultaneously in order to manage DNA tangles and supercoils. Lastly, the topoisomerase 4 transport one segment of DNA through a transient double-strand break in a second segment of DNA. Step 2 is primer synthesis. A primer must be synthesized by an enzyme called primase, which is a type of RNA polymerase, before DNA replication can occur. The synthesis of a primer is necessary because the enzymes that synthesize DNA, which are called DNA polymerases, can only attach new DNA nucleotides to an existing strand of nucleotides. Primase is an enzyme that lays down RNA primers which enable DNA polymerase III to make DNA. And then the DNA polymerase III comes in and he needs that 3' and OH from the RNA primers in order for it to continue to build nucleotides. Enzymes known as DNA polymerases are responsible for creating the new strand by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria, such as E. coli, polymerase III is the main replication enzyme, while polymerase I, 2, 4, and 5 are responsible for error checking and repair. In eukaryotic cells, the polymerase alpha, delta, and epsilon are the primary polymerases involved in DNA replication. The elongation phase of replication includes 2 distinct but related operations. We have the leading and the lagging strand synthesis. New nucleotides can only be added to the 3' end of the existing chain. In the leading strand, it has its new strand continuously synthesized as helicase unwinds more DNA, while in the lagging strand, it is a discontinuous replication because the bases are oriented in the wrong direction. The DNA polymerase can only add nucleotides in a 5' to 3' direction. The leading strand synthesis begins with the synthesis of RNA primer by DNA polymerase at the replication origin. The leading strand synthesis then proceeds continuously in the direction of the replication fork movement. Lagging strand synthesis is synthesized discontinuously in the opposite direction from the replication fork movement as a series of small fragments known as Okazaki fragments. It was Reiji and Junichi Okazaki who discovered the Okazaki fragments in 1968. Each Okazaki fragment is then initiated by the synthesis of RNA primer by polymerase alpha and then completed by the synthesis of DNA by DNA polymerase delta until reaching the next RNA primer. Both leading and Okazaki fragments of lagging strands are synthesized from 5' to 3' direction. And as we know earlier that there is a leading and lagging strand for each of the two replication forks. Then the DNA polymerase epsilon begins to remove the RNA primers. Both DNA polymerase delta and epsilon have the ability to proofread their work by means of a 3' to 5' exon nuclease activity. If DNA polymerase makes a mistake during DNA synthesis, then the resulting unpaired base at the 3' end of the growing strand is removed before synthesis continues. So this is an animation showing the leading and lagging strand in DNA replication, and as you can see in the leading strand that it is replicated continuously by the DNA polymerase because DNA polymerase builds a strand that runs anti-parallel to it in the 5' to 3' direction. And on the lagging strand, DNA synthesis restarts many times as the helix unwinds, resulting in many short fragments called the Okazaki fragments. That there is a ligation of the newly synthesized DNA segments with the help of DNA ligase or glue enzyme, which seals the nicks between the Okazaki fragments, converting them to a continuous strand of DNA. DNA ligase requires ATP to form a phosphodiester bond between the two nucleotides. Termination of DNA replication is less well understood than initiation and elongation. Genome duplication is carried out by pairs of replication forks that assemble at the regions of replication and then move in opposite directions. DNA replication ends when converging replication forks meet. So at the end of DNA replication, the RNA primer are replaced by DNA by 5' to 3' exon nucleus and polymerase activity of DNA polymerase epsilon. Exonuclease activity of DNA polymerase removes the RNA primer and polymerase activity adds DNTPs at 3' OH and preceding the primer. Bacteria with circular chromosome do not have this problem since their replication terminates only when two replication forks meet one another at a designated termination site. But in eukaryotic organism with linear DNA, there is a problem. When RNA primer at 5' end of daughter strand is removed, there is not a preceding 3' OH such that the DNA polymerase can use to replace by DNA. So at the 5' end of each daughter strand, there is a gap or a missing DNA. This missing DNA causes loss of information contained in that region. This gap must be filled before next round of replication. For solving this end replication problem, studies have found that linear end of DNA, called telomere, has G and C reach repeats. This sequence is known as telomere sequence. These repeats of telomere sequence is different among different organisms. Telomere in human cell consists of repeats of TTAGGG. Each species has its own specific telomere repeats. These telomere sequence do not code anything, but it is essential to fill the gap in daughter strand and maintain the integrity of DNA. There is an enzyme found in eukaryotic cell called telomerase. Telomerase is a DNA polymerase which adds many copies of telomere sequence at 3' OH end of template strand. Unlike other DNA polymerase, telomerase adds DNA at 3' OH end of parent strand, not at the daughter strand. And also, it synthesizes the same sequences over and over in absence of template strand. The figure here is showing that the end of the chromosome is faithfully replicated. First, telomerase binds to 3' OH end of parent strand by hybridization between its AACCCC AAC RNA sequences and TTGGGG DNA sequences. The telomerase adds TTG at 3' end of the parent strand. After adding TTG sequences, telomerase translocates along 5' to 3' end of parent strand. Now, the telomerase adds GGGTTG to 3' end by using its CCCAAC sequence. Again, telomerase translocates and adds GGGTTA sequence. This process is continued for many times. The parent strand becomes more longer than a daughter strand. Now, RNA polymerase, or the primase, synthesize RNA primer by copying the parent strand in 5' to 3' direction using telomere sequence as template. Next step is the DNA polymerase can now extend the primer in 5' to 3' direction by adding deoxyribonucleotide to 3' end. Then, the primer is now removed and it won't be replaced because it is an extra sequence added by copying the telomere sequence. Finally, the integrity of daughter strand is maintained. And that is how DNA is replicated. Trivia. The typical human chromosome has about 150 million base pairs that the cell replicates at the rate of 50 pairs per second. At that speed of DNA replication, it would take the cell over a month to copy a chromosome. The fact that it only takes 1 hour is because of multiple replication origins. Now let's proceed to the comparison between the DNA replication of prokaryotes and eukaryotes. Prokaryotic replication occurs in the cytoplasm while eukaryotic replication occurs in the nucleus. Prokaryotic replication happens during the first step in binary fission, while the eukaryotic replication occurs during S phase of cell cycle. There is only one region of replication in the prokaryotic replication and multiple regions of replication in the eukaryotic replication. There are 100 to 200 nucleotides in the region of replication in the prokaryotic replication and 150 nucleotides in the region of replication in the eukaryotic replication. There are 2 replication forks in the prokaryotes and several replication forks in the eukaryotes. There is only a single replication bubble in the prokaryotes and several replication bubbles in the eukaryotes. There is one replicant in the prokaryotes and multiple replicants in the eukaryotes. And the initiation in the prokaryotic replication are by the following proteins, DNA A and DNA B. And the initiation by multi-subunit protein or region recognition complex are in eukaryotic replication. So here is a table showing the difference between the prokaryotic cells and eukaryotic cells in terms of step in replication. So here are the differences. The origin of replication in prokaryotic cells happens in one per chromosome, while in the eukaryotic cells it has multiple per chromosome. In synthesis of DNA, it has DNA polymerase 3, while eukaryotic cells has DNA polymerase alpha and delta. In removal of RNA primers, prokaryotic cells has DNA polymerase 1 and eukaryotic cells has RNase H. In the replacement of RNA with DNA, prokaryotic cells has DNA polymerase 1 and eukaryotic cells has DNA polymerase delta.
In the synthesis of telomeres, it is not applicable in prokaryotic cells, and in eukaryotic cells, it has telomerase. So here are some figures showing the difference between the DNA replication of eukaryotic cells and prokaryotic cells.
Thank you so much for listening.